Welcome to zoology session. Today's topic neural control and coordination. In that we will discuss generation and conduction of nerve impulse. Nerve cells are excitable. It shows one of the property of a neuron. Excitability electrically charges reaches to target place to through the nervous system, nerve fibers. So, suppose this is the neuron. Here, this is cell body, dendrites, and a nucleus. Here this is the axon covered with myelinated, and this is the node of Ranelagh. Here when stimulus started, this stimulus travel through the nerve fiber and ends with release of neurotransmitter through action potential, that is called as impulse. Impulse generates through action potential. So what are the various types of stimulus, external or internal, to generate this impulse? We will discuss. First one, physical. Physical stimuli due to the heat are cold. Chemical stimuli, acid, base. Electrical stimuli, plus minus charges. Mechanical, that is due to injury. These are outside, and inside also stimulus response takes place. If stimulus started here, it passed through the nerve fiber and ends with the synaptic knobs. Here neurotransmitters releases. Though this process called as impulse. This impulse is nothing but action potential. So how it is taken place? This nerve impulse have 4 states. Before going to that, what is axolemma? What is exoplasm? What is extracellular fluid? What of the various ions are present in this? And due to that, the diffusion of the ions, different ions, Na plus, K plus, Cl minus and the Ca plus, how it is exchanged through the membrane and how it generates the impulse in a exoplasm, how it conduction takes place, we will discuss in this topic. If this is a cell body and here it is these are, this is the nucleus. Here it is dendrites, which takes place stimuli here through the nerve fiber. This portion indicates axon. So this is the axolemma, that is membrane. This is also axolemma. axon membrane. Inside of the axolemma there is a fluid, that is axoplasm, and outside of this membrane or axolemma extracellular fluid present. This is extracellular fluid, and inside this is axoplasm. This is the axoplasm. So this is very important to understand. Why? Because of when action potential generated, outside of this extracellular contain sodium plus ions, and intracellular, it means inside of the membrane there is K plus ions are present. There is a difference between the ions due to compensate of differentiation. Inside and outside the ions may be exchanged takes place through the channels. Those channels are present in a axolemma, either in the dendrites and here also. Those channels we differentiate three types. One is leakage channel. So this leakage channel allows K plus and Na plus ions through the membrane. These leakage channels are present in a dendrites area and axoplasm area. So this membrane contain leakage channels, sodium and the potassium. Potassium are more channels present. Due to that, membrane or axolemma allows more potassium into different places. Next one is ligand-gated channels. These ligand-gated channels are present here post-synaptic. This is one of the dendrite, and another one more neuron receives the signal. Like here it is, one more dendrite connect with the presynaptic nerve, and this we call as a postsynaptic. Here there is a neurotransmitters are present. That neurotransmitters allows through the channels. So ligand-gated channels are present. Cell bodies are dendrites, and it allows neurotransmitters are chemicals. Chemical signals. Third one is voltage-gated channels. These voltage-gated channels are present here. This is extracellular fluid, and here it is axoplasm. These membrane contain voltage-gated channels. That is, I will draw here. This is these two are membranes, indicates green color portion, and here this is the neurolemma, axolemma, and between this there is a plasm, fluid filled, that is axoplasm. It is axoplasm, and it is axolemma. So this portion I am giving here, and out of this axolemma, either side extracellular fluid is present. So there is two types of channels. One is Na plus and K plus. Na plus channels again activation channel and inactivation channel, and K plus is only activation channel. Now I will tell you, here there is Na plus channel, outside of this neuroplasm, axolemma, extracellular fluid side, this is activation channel of Na plus, and either side, inside, this is inactivation of Na plus channel, and here it is one more channel, that is K plus, only activation channel. So where there is a ion diffusion takes place due to the concentration gradient of the ions, and these ions exchange through diffusion or gated channels outside and inside and balance, and it potentiate, it generates the plus minus charges difference. That difference we indicates, suppose these are the axolemmas, and if these axolemma either side plus and this is extracellular fluid and exoplasm, that is negative charges, both sides, not only one side, both sides, here it is positive and the negative. So these two areas potential, whatever negative charges are difference, that we called as potential difference. So this potential difference generates the action potential, and it conducts the nerve impulse one place to another place. In this nerve impulse, there are 4 states we will observe. So, nerve impulse, states, various states we will discuss in this topic. So, first state is resting state. Previously we discussed, this is the axolemma and the sodium gated channels, potassium gated channels, and here extracellular fluid, this is the exoplasm. In resting state, plus or positive charges are in an extracellular fluid side and negative charges are in exoplasm. And here sodium ions are present and here potassium ions are present. In this state, inactivation, inside of a inactivation sodium gates are open and outside activation gate sodium gates are closed, and K plus sodium K plus gates also closed. And after this, when stimulus taken place, there is a change, diffusion of ions taken place, and between this area and the positive area and the negative area, there is a difference between the charges. That already we discussed, potential difference. That potential difference indicates minus 70 millivolts at resting states. Here, this state we call as a polarization state. So here what happens? There is a ion exchange takes place through the plasma membrane, and it reaches to when 3 sodium ions enters into exoplasm, 2 K plus ions exit or efflux from exoplasm. So it enters into the extracellular fluid. Due to the difference, this sodium pump and the potassium pump uses ATPase and diffuses through the membrane. What happens? This is polarization state. Again, sodium activation and inactivation gates both are open. Sodium enters into the exoplasm. Due to that, here more positive charges takes place. Due to that, outer side of exolemma, more positive charges increases. So if observe resting state, there is a inside positive charges, outside of the membrane negative charges. This state we call as a depolarization. Inside the membrane indicates positive charge and it reaches to plus 45 millivolts. And here, whatever the positive charge takes place 45 up to 45, this position we call as a spike potential, and also we call as a positive feedback regulation. More positives we call as positive feedback regulation. And later on, after this state, repolarization, when positives, sodium and the activation and inactivation, both gates are open, and remain here potassium gates closed, here also potassium gate closed. Due to that, K plus do not move from the exolemma to extracellular fluid. In repolarization state, here K plus ion channels open and the efflux of K plus enters into extracellular fluid, and Na plus also moves outside of this, and here inactivation gates, inner side of the gates closed, and activation of Na sodium open. Now more positives, K plus positives are entering into this region. Again, how the resting position state, out of the exolemma, positive charges is more and negative charges inside is more. That's why this we call as repolarization. So after this state, again hyperpolarization. Hyperpolarization, when it takes place, more sodium enter into the extracellular fluid, becoming more negative inside of the exoplasm, and due to that, more minus or negative charge become minus 90 volts. So being resting state have minus 70, more than that the minus 90 volts, millivolts reaches a spike undershoot condition. So beyond this resting state, that's why this state we call as an undershoot state. So this is the excitable position, and here also excitable position. Here lastly sodium and potassium channels are closed, but here activation of a sodium open and last sodium slowly closed, and later on total charges are remaining, it comes into the resting state. So this is a cycle of nerve impulse, and one state to another state it excited and takes place the action potential, and these potential impulse travel one place to another place of the nerve fiber, and this we called as a generation and conduction of the nerve impulse. In this we will observe refractory period. In this refractory period there is no generation, another stimuli conduction do not takes place while generating the action potential. That's why this we call, this being of this period we call as a refractory period. So this refractory period, two types, that is absolute and relative. This absolute refractory period indicates hyperpolarization and depolarization situations. Here there is no another impulsion takes place when the action potential is generation taking, and relative refractory period we will observe in a hyperpolarization. So absolute depolarization and repolarization both we will observe. Depolarization and repolarization. And a relative, there is another initiative impulse start with the another generation. That's lastly hyperpolarization. And we will discuss middle of the graph. First, if we observe there is a graph, we know that there is a zero above indicates the positive charge, and the below zero minus charge. Here 0 below we will observe minus 70. This minus 70 millivolts of energy indicates resting state, and it continues when it reaches to minus 55 millivolts, that is reaching to the threshold stage. And after this reaching threshold state, more positives, increasing of more positives, again above the zero it reaches to 45 plus 45 spike potential and a positive feedback regulation. After that again it reaches to the negative state, that is before going to negative state repolarization, there is a plus 20 MV millivolts energy is present in this, and hyperpolarization minus 70 indicates here, below that there is a minus 90 millivolts energy showing here, and later on again it comes to the minus 70 MV. Like the generation how it is takes place due to the graph we can remember, and once we can recall what are the gates are important when which state indicates, and when the gates are opened ions how it is exchanging. Now first we will discuss resting state. Here K plus closed and Na plus active channel closed, inactivation channel open. Depolarization, here activation of sodium, inactivation of a sodium channel open, and K plus channel closed. If you observe the repolarization, all three of these channels are open, and hyperpolarization, here inactivation channel is closed and activation channel is open, and a K plus channel open remain, later on it also closes. then it goes to the resting state. So this is the nerve impulse, how it conducts. The mechanism already we discussed.